So I'm Martijn Verdoes, and I'm at the Radboud University Medical Center at the Department of Tumor Immunology. But my background is in organic synthesis, actually, so in pure organic chemistry. So chemistry is a science of connecting. And now with my group, we're trying to connect basically two disciplines, being chemistry and immunology. And I'll hope to show you why and how we do this. Let's go to the next slide. So obviously I didn't do all the work, and this is the group that I'm currently have the pleasure with and the privilege to work with. These talented PhD students and former postdocs have mainly done all this work, and some of it has been done with the Department of Nuclear Medicine, with the group of Sandra Heskamp, and also with the group of Vera Scheren at Leiden University Medical Center. And I also want to obviously acknowledge the funders who made all this work possible. So most of you know cancer as a disease that comes about due to uncontrolled division of derailed cells. But less people actually are aware of the fact that cancer is not just cancer cells, but more sort of a diverse combination, or almost city, let's so to speak, of a bunch of different cell types. And the tumor, the composition with all these different cells, we like to name tumor microenvironment. And what's actually apparent is that in a lot of tumors, almost half of the tumor mass can actually be composed of immune cells. So these are the cells that are basically there to protect your body from invading bacteria or toxic materials, but also from cells that actually become derailed, and they need to be eliminated by your immune system. And this luckily happens in most of the cases, in several occasions daily in most individuals. But when the combinations are right, or actually wrong, when the hallmarks of cancer are met, then a tumor can actually arise. And these tumors, they attract all kinds of different cells with different traits. So and lots of immune cells, they have the ability to migrate. And therefore, it's not surprising that actually these immune cells can help the tumor to migrate themselves. So they are their proteolytic activity, their ability to migrate, they help tumor cells to, for example, form metastases. But also, a lot of surface molecules, a lot of the traits of these immune cells are exploited by a tumor to later then evade the immune system. Because the tumor, the immune system is always in a balance between tolerance and immunogenicity. And tolerance is important, and you all might be familiar with the problems with transplantations. So things that are self need to be tolerized, so not be attacked, and things are a danger, potential danger, and from the outside need to be attacked. And the tumor is actually sort of in the middle. And the tumors, they exploit surface molecules, but also create chemical environments, sort of pH and oxidative states, to basically protect the tumor site from eradication by the immune system. And one very important mechanism that has led to a breakthrough in cancer therapy is actually the fact that when you have a cytotoxic T cell, which is an effector immune cell that has a job to kill either virus-affected cells, but also cancer cells, it functions through the recognition of a peptide that's presented by a cancer cell through the MHC1 complex, and is recognized by a T cell receptor. And when this recognition takes place, the T cell gets activated and then secretes all kinds of nasty stuff into the cancer cell that makes it die. But to generate tolerance, the immune system also has counteracting mechanisms. And one of these examples is programmed death 1 and programmed death ligand 1. And this is sort of acts as an immune checkpoint, as we call it, and it's basically a break on the immune system. So when a cell expresses this ligand, is then recognized by the receptor on the T cell. This then thinks, oh, this is a self cell, for example, therefore I want to kill it. And cancer, as I mentioned, exploits this mechanism. And antibodies, so a class of molecules that you are all aware of in the context of COVID, obviously, is these are a class of molecules that are super specific in recognizing certain molecules. For example, surface molecules, and you can engineer those. You can generate and engineer those to be used as a therapy. And these checkpoint inhibitors, they basically break the brake of the immune system and thereby allowing these sort of dampened cytotoxic T cells to then again recognize the cancer cells and kill them. And as mentioned, this led to a breakthrough in immunotherapy. But unfortunately, although cancers that were previously untreatable are now treatable, only a minority of or a fraction of the patients actually benefit from this treatment. And therefore, and this is just one of the molecular strategies that we can use to design therapies. So, as mentioned, the tumor actually is tilting the balance towards tolerance, and using chemistry or chemical immunology, as we call it, we want to design and synthesize new molecules to study the role of the immune system in the context of cancer, but then ultimately to use these concepts to then basically tip the balance away from tolerance towards immunity. And how we like to approach it in my lab is to, at the one end, try to boost an anti-tumor immune response by targeting specifically dendritic cells, which is a type of immune cell that helps, that basically teaches the immune system to recognize what's wrong. And at the same time, to break the immunosuppressive barrier that's present in the tumor microenvironment to actually allow the tumor-specific effector cells that we want to generate with the first approach to be able to actually enter the tumor to do the job. So precision immunotherapy. Precision comes by targeting in our case, and we use several kinds of molecular entities to target the different aspects. So as mentioned before, we can use antibodies, and we have developed certain strategies that allows us to specifically modify these antibodies so that we can sort of deliver cargo to a place without destroying the capabilities of these antibodies for recognition or activation. And we use also chemokines and other single domain antibodies, for example, all the way through polymers, also to small molecules that sort of exploit the chemical environment present in a tumor microenvironment. That can be pH, reactive oxygen species, but also protease activity, for example. Proteases are enzymes that degrade other proteins. And this is sort of an overview of some areas that we're currently working on. So we're trying to develop new types of dendritic cell targeted cancer vaccines, and I'll get into that in a little bit. But also we use antibodies and antibody fragments to deliver multiple components to one site to serve as diagnostics for multi-scale purposes, but also for theranostics, which is a combination of diagnostics and therapy. And as mentioned, we're developing new types of strategies to deliver either cytotoxic or immunomodulatory cargo, small molecules, to the sites of a tumor to, for example, eliminate certain immunosuppressive immune cells that are present at the site of the cancer. But these immune cells are also very plastic in their activation state. So you can actually push them, so you can turn the bad guys into the good guys, so to speak. And as I mentioned, in some tumors, it can be up to 50% of bad guys. If you can turn them all into good guys, that can give a lot of therapeutic benefits. So as mentioned, this immune checkpoint therapy, this antibody-based therapy, works for a bunch of patients, but not for all. And PD-L1 expression, so the surface marker on this cancer cell, has been used as a biomarker to try to predict therapy outcome. But conflicting correlations have been found. And the reason for this can be found as we zoom in a little bit on the tumor. Most of these biomarker analysis has been done on biopsies. So you take a little bit of a tumor and then look whether PD-L1 is present or not. But PD-L1 can be present on the cancer cells themselves, but also on infiltrating immune cells. And these tumors are actually very heterogeneous. So by taking a biopsy, you basically get a sort of a snapshot, which might not reflect what's actually going on in the rest of the tumor. And therefore, we try to think of a strategy which allows us, on a multi-scale level, to determine if, and if so, where exactly in the tumor and on which cell types this PD-L1 expression occurs. And in order to do so, we want to take a therapeutic antibody, like the checkpoint inhibitor, but then equipped it with imaging agents that allows us to track it on different scales. And in order to do so, we recently published a strategy where we use CRISPR-HDR and genomic engineering to turn hybridoma cells into cell lines that express and produce high amounts of antibodies or antibody fragments in which we can change the type of the antibody, but also introduce specific sites where we can introduce different types of molecular entities. And we use this then to, so here's an example. So we take an anti-PD-L1 antibody that's therapeutic in mouse models of cancer. We engineer it to then become a mouse antibody and to be equipped with certain tags that allows us then to introduce a synthetic peptide in which we have a molecule which is highly near-infrared fluorescent, and we have a chelator to introduce a radio ligand, a radionucleus for imaging purposes. And nuclear imaging allows you on an entire body scale to localize regions where the agent accumulates, but the resolution isn't very high. But that's why we introduce a near-infrared dye, because for this the penetration depth is not so great, but the resolution is super high. So then we site-specifically equip them with these two cargoes, and then here's an example where we have a syngeneic mouse model of breast cancer. And we can see that this tracer now accumulates based on fluorescence in the tumor region, and also with SPECT-CT, we can find accumulation of this checkpoint molecule in the tumor, but also in immune-related organs. Sorry?
Lo que hacen por nosotros.
I think someone forgot to mute themselves. So, but the nice thing now is that we can also already see, if we take out the part of the tumor, we can see that it localizes heterogeneously throughout the tumor. And if we then look at the microscopic scale, we can actually now, on a cellular level, determine where the tracer and thus also where the therapeutic ended up in this tumor. So another example is that we try to boost the immune response by generating new vaccines. And a classical vaccine basically delivers antigen and adjuvant, for example, in a needle or a drop or a bubble, as we heard before. And then this antigenic cargo needs to be taken up by an antigen-presenting cell, which then goes to the lymph node where it finds T cells, and where then these T cells get activated to do their job. At least that's the theory. But for a proper activation to occur, you need the antigen to be presented, but the antigen-presenting cell also needs to be activated, because if this is absent, then it actually tolerizes the T cells rather than activating them. And this is a problem because the antigenic cargo has totally different pharmacokinetic properties than the adjuvant, meaning that if you administer it or inject it into a subject, the antigen will go one way and the adjuvant the other. And they need to go to the same place. So we're trying to develop strategies that actually allow us to deliver the cargo, the antigenic, as well as the immune stimulatory cargo to the exact same cell. and even the exact same endocytic compartment in a certain cell. And we use nano vaccines for that, but also single molecules. And we use a similar strategy. Sorry.
Starting to wrap up now, please.
Yeah.
Yeah.
So I'll skip right through it. Long story short, we can synthesize these peptides. We can fuse into these antibody fragments that can deliver it to these DCs. And then we can also conjugate to this exact same construct the immune stimulatory component. And this is just proof that we actually made it. And if we then have a co-culture with these dendritic cells and these T cells, if we administer the antigenic cargo targeted to a dendritic cell, we can already see that, and this is a measure of T cell activation, that is increases. But if we then co-deliver this immune stimulant, this doubles. And if we take an antibody fragment that is not recognized, that does not recognize something on the surface of this dendritic cell, then we don't see this activation. So a nice molecular trick to co-deliver stuff to an antigen-presenting cell to give proper immune cell activation. So this is actually a slide to wrap up. So it was nice to show you a couple of examples of two strategies that we're using to monitor and try to manipulate the tumor microenvironment at the one end, and at the other end to try to boost the immune system to recognize and to eliminate cancer. Thank you very much.
Thank you very much, Martin. I see already that David has a question. We're a little bit over time, but this is usually what happens. So we still have, if we say we have 4 or 5 minutes for questions. David.
So I was just interested in knowing, so you said vaccine and you make allusion to bottles, so we definitely need to talk separately.
Yeah.
But so how easy is to translate? I'm assuming you have some sort of animal models, but you first test a lot in vitro in the lab.
Correct.
So how important is, how to say, this sort of step between in vitro and the animals? Like, do you need to do something extra, or is just getting things in an ampoule and then injecting directly?
So actually the vivo stuff, unfortunately, is very important because immune cells, as I already mentioned, also in the context of the cancer, they are highly plastic. So when you take cells out of their native environment, they change, they respond. So if you take a cell line or take cells and treat them ex vivo, they're basically different cells than you would encounter in vivo. And also, I think, so we try to do most of the stuff in vitro to get a feel, especially when we have different sets of and different types of designs of molecules, which we expect to then in the end to perform best in vivo. But also in relation to what I mentioned for the sort of the classical vaccination, it's super crucial to test the behavior of these large constructs actually from a molecular perspective, how they behave in vivo, how they distribute, how they get cleared, how the off-target effects are. And unfortunately, this is still best done in mouse models. Yeah. And then, so for example, so we're now in the middle of a clinical trial where we're testing the first generation of these nano vaccines in human. And for that, obviously ample in vitro and mouse model experiments have been done. And based on that, we were able to actually get phase 1-2 trial approved. Yeah.
We do have time for one more question. There's never enough time for models. Any more questions from anyone? Then I think we'll start to wrap things up. Again, I learned a lot from the 3 talks today. One of the things I learned was that I miss a photo with my research team holding props like bowling balls or bubbles. So my team looks very boring. I'm just holding books or something. So I especially like the bowling ball being balanced over top of this poor girl's head, and she doesn't know it.